Host, can you please enable, permission to share my screen, please?
Yes, sir. Yes, sir.
Once you grant the access, let me know so that I'll start sharing my screen.
Sir, should try to screen share.
So hope everybody able to see my screen, right?
Yes, sir.
Yeah. So shall we start, or we'll wait for some more time for to join other colleagues, like other students?
Other colleague, if it is
So, good morning, one and all. A warm welcome to Dhanvantri Institute of Pharmaceutical Sciences webinar Pharma Talk 3.2. So, coming to about speaker, today our chief guest, Dr. Amarnath Reddy Garu, Deputy Manager, Pfizer India Limited, and he has 10 years experience in parenterals development, and presently he is associated with Pfizer from the past 5 years, and he had hands experience on making of dry powder parenterals, lyophilized products, and liquid injections, on market support. And now we cordially invite Amarnath Reddy sir to continue his section of Pharma Talk 3.0, that is development of sterile dosage forms to the regulated markets. Sir, please continue your section, sir.
Hello. Shall I start?
Sir, please begin your section, sir. Yes, sir. Yes, sir.
Yeah. Thank you. Give me a minute, please. Yeah. Good morning, one and all. Thanks for giving the wonderful opportunity. It's been long time. I was interacted with college students. I had completed my master's in 2012. After that, I never interacted with any students, and it was a wonderful opportunity to me. Dr. Ravikant told that you give a session to the students on the sterile injectables, and my sincere thanks to Honorable Dayakar sir, who is the principal, and other event directors and event coordinator Dr. Nagaraju sir, and other organizing committee members who are in regular touch with me, to understand this presentation, and whom mobilized this activity. Thanks again once. Thank you all. Let's start the session. If you guys had any point of time, any doubt, please stop me. That is fine, because I feel more comfortable in that way, rather taking the question and answers, clarification at the end, because if at all any if you miss the point there, if you are going beyond that, we will miss that loop. So I always prefer, whenever you have a doubt or anything you need more clarification, please stop there. So we can have a discussion there, and we can move forward. And one more thing, consider this as consider me also another student, because we in a industry we always learn, like from the day to day, our activities in the labs, whenever we are doing it. Make it more interactable, interactive session. Anytime you just want to understand anything, please let us know. And even we also want to learn so many things from you guys. Yeah, thank you. So let's start the session. And today we are going to discuss about research and development of sterile injectable. The reason why I had selected these sterile injectables is basically I am working in sterile injectables from the last 10 years. 2012 I had completed my M.Pharm. After that I had worked in 4 different companies. Presently I'm working at Pfizer, Chennai R&D. From the beginning I was into sterile injectables. I didn't work in solid orals, basically. Only I have a subject knowledge, how you guys having what we learned in B.Pharm and M.Pharm. Sterile injectables I have comparatively better idea, since being working in that platform. And you can see there were some pictures, your right hand side. All these were comes under the sterile injectable technologies. This you might be knowing, like ampules. This is a clear ampule and this is a amber ampule. Are you able to see my screen and cursor as well, right?
Yes, sir.
Yeah. One more thing is, these are called vials. You might be seeing in day to day, whenever you take any pharmacy store or something. These are called vials, and these are called pre-filled syringes. These are little advanced drug delivery systems. It's a drug delivery system. That's all. Consider the same drug solution. You can keep it in ampule, or you can keep it in a vial, or you can keep it in a pre-filled syringes. And then again, so in pre-filled syringes, there were huge advancements are happening time to time. You can see, like this contains needle. This is called stacked needle pre-filled syringes. So which means that the needle also will be there inside to the pre-filled syringe, so that nothing, you can directly administer to the patient, like whenever you want to give. Whereas in this you can see it doesn't contain any stacked needle, but it was closed with a end with one more rubber closer. This is also called as a rubber stopper. So all these are called as rubber stoppers. The backside one will be called as plunger stopper because it was connected to a plunger, like your regular Dispovan syringe. If you see, the plunger which is attached to a stopper is called plunger stopper. Then this is called barrel, pre-filled syringe barrel. Inside that our drug solution will be there. Again, this is called lure lock tip, which means that it to close the pre-filled syringe so that the solution should not come outside. Basically, by principle, it won't come out, but still in a safer side, we will keep it. So whenever doctor wants to go for it, what they will do, they will take out this lure lock tip, then they'll attach a needle, then they will give it. The interesting thing here is, I just want to illustrate one more time here. This is the advanced drug delivery than the stacked needle. What happened? This needle is made up of tungsten. So what, like initially it was a very successful drug delivery system. What happened over a period of time? There was so much of impurities. What you can say, like you can consider active pharmaceutical ingredient is 100%. You consider there will be some byproducts will form. In pharmaceutical terminology, we will call it those who are as degradants or related substances or impurities. So what happens is, this stacked needle, whatever tungsten is there, this is interacting with your drug solution. Over a period of time, it is leaching some metals into the drug solution, which is a serious concern there. So that's why later the evaluation was happened. They removed that entire needle and they kept a stopper. Again, you can ask one more question here. Yeah, stopper also can give, it's a rubber material, right? So this stopper also will give some more impurities, but whatever the leachables, like stop from the stopper, which is coming inside by in contact with the drug product solution, having well established literature data, maybe from the academic perspective, some students like you guys might have been generated what is coming inside from this stopper. And so that's why this is called as advanced drug delivery system. Then you can see here, these are some other drug delivery systems which you can see. If someone is taking insulin pen, there were very advanced drug deliveries are there, where like a meter dose, which means it will deliver exact amount of dose in each particular time. We all might have read in BPharm. Then again, this is Pfizer's patented drug delivery system. These are called Abosect syringes, and this is called plastic answer syringes. So then today, any point of time, if you guys want to have little more discussion, please stop me there. Like we can have a more interactive session. So today we are going to discuss about dry powder parenterals, which is a sterile powder fill and lyophilized product, and liquid injectables and complex injectables. So dry powder parenterals are nothing but which contains basically, I don't want to go to what is the injection and all. Whenever you see any injection formulation or any dosage form, if you see, it contains one or more active pharmaceutical ingredients. May or may not excipients will be there, right? So dry powder parenterals are nothing but what happens here is, in a sterile environment, the API or mixture of API with the excipients will be filled into a vial in a aseptic environment. Then that will be called as a dry powder parenteral. Then lyophilized drug delivery, consider you don't have a aseptic facility, making dry powder is not an easy task. Sometimes your API may not be stable, for which you may need to have some additional excipients, like for example some buffering agents you need to add it. as we know that some products will be stable at particular pH only. Okay, to give that buffer strength, we need to add some excipients in terms of like, you know, buffering agents, or some excipients which will give enhanced stability to the drug product. So in such cases, what we need to do is we will go with the lyophilized approach, where we will make a liquid solution, like in a water or suitable vehicle. We will dissolve all the API and excipients, then we will lyophilize it. Then liquid injectables will be two types. Liquid injection, whatever you had made the liquid injection, it can be aseptic process, which means that you can filter through a filter, aseptic filter, then you can give to the patient. Or if the product doesn't have any thermal degradants, which means that some products was not stable at heat, right? So such products you cannot apply them terminal sterilization, which is nothing but your autoclaving. Like simple terminology, I'll say 121 degrees centigrade, 15 minutes, 15 minutes, what we will do autoclaving. In that way, you can do the terminal sterilization. Remember, always as a pharmacist, you should go for the terminal sterilization, because you have to put all your efforts to make it happen. Sometimes you cannot do it, but you have your approach should be to do the terminal sterilization, because when you do the autoclaving, it will be 99.99% the whatever the spores or organisms will be there inside that was killed during the autoclaving. Whereas in the aseptic process, only thing is you are filtering. What about, like, for example, consider your filter was not integral, which means that if your filter having some holes, or if the filtration is not proper, then you gave a non-sterile injectable drug into the patient, which leads to fatal, like, the patient will die. There is no second thought on that, because that's what the injectables, the advantage of injectables is rapid onset of action. The disadvantage is you cannot take out, because it will leads to a fatal, immediately it will enter into the systemic circulation, so that which you cannot take it. But as a pharmacy students, always when you go into the pharmaceutical industries in the coming days, always you have to question yourself, why can't I do the terminal sterilization? Maybe some heat degradants might have going above the threshold limit, but try to identify what is that impurity, what is the degradation is happening, and how far that degradant is not toxic. There will be we know that there will be so many impurities will be there in any dosage form, but only thing is it should be in a acceptable level. If you want to go beyond the specified guidelines about values, you have to do a qualification, like you have to isolate that impurity, then you have to do the NMR mass spectroscopy, identify it, and just like as I said that always we'll go for the literature which was published by you guys, like students, whoever publishes. Like, for example, I got a heat degradant of A. I had I did a mass spectrum, I know the mass number, and I did the NMR. I did the complete characterization. I know the IUPAC name of that particular impurity. Then always I'll search in the literature, like you guys might have published some literatures. Is there any other formulation this impurity was there, whether that's a toxic or not? If at all, if it is a toxic, which means if it is not acceptable to inject into a patient, how far I can do it, like what level I can do it. For example, it can be up to 1% you consider, but whereas when you are doing the terminal sterilization, you are getting 1.1%. It's a failure, but you have to put efforts by doing some toxicology studies, which we had read in pharmacology of B Pharm. Toxicological studies we will do, preclinical followed by clinical studies, where certain rat mice we will take and we will inject that particular impurity into them, and then we will establish the toxicological profiles. The reason why I am stressing so much here is on the terminal sterilization. People will, I worked in different organizations. Some of the people, what they will do, filtering is a easy process, whereas terminal sterilization is a big process. People don't want to go for a robust approach, which is a terminal sterilized, which is damn well established, sterile, sterility assured product. They will go directly to the aseptic by saying, yes, my product is failed and heat degradants are going beyond the threshold. But I'm saying, you guys, when you enter into the industries, always put efforts. Why can't I, establish a little more level of toxicological data to that particular impurity? Then complex injectables, emulsions and suspensions and liposomals. And one of the examples, liposomals, you can see there was a black fungus is around in the news, you might be heard of, for which amphotericin B liposomal injection is the drug delivery which they will give for this fungal infection treatment. Okay. So this is the evolution I just want to tell, because can you minimize, please, whatever is there on my right hand side? I'm unable to do it. Okay. I'm getting a separate pane with the list of people. Can someone do it? Minimize it, please. Okay. Okay. So the reason why I just want to tell the evolution is, since you guys, maybe a year or 2 years or down the line of 5 years, you may enter into the pharmaceutical industry. Always you have to be ready for the change, because you should not say that still I'll use the ampule to inject into a patient. Okay. So it is a very dynamic world. Things are going very rapidly. And there were some advantages also when you go for, evolution. For example, if you see earlier what they used to do, ampule they used to break, they used to take out the drug solution from there and they used to inject it. Okay. But if at all you consider the first picture which I had kept in the ambered color vial, Trianda for injection, where the product is a lyophilized vial, which means that the product is in a lyophilized form. So you cannot give a solid powder into intravenous or intramuscular or subcutaneous, injectable way. So for which you need to do a reconstitution, which is nothing but you may have to add a appropriate water for injection or 0.9% sodium chloride injection or whatever it is, appropriate diluent you have to inject into that vial. Then you have to do the reconstitution. Then you have to take out that solution from the vial. Then you have to administer it to the patient. You just see how many series of steps involved. Consider you don't have a WFI vial, water for injection or diluent vial. You cannot give to the patient ultimately. Then you want to dilute with the 10 ml, but whereas you have a 5 ml WFI, what you do? You cannot do it. To say these are the very dynamics which are involved. Why the industry is going in such a rapid way, in a dynamic way, to advanced drug delivery systems. Then what if you have to reconstitute with 10 ml, but in a faster manner, the nurse might have only taken out 10 ml and she might have instead of 10 ml, she might have reconstituted with 8 ml. So by which you are giving more concentrated drug to the patient, which was not clinically safe to the patient. Okay. And possibility of medication error is very high in this dry powder parenterals or lyophilized product. Consider next drug delivery. And the reason why I had kept this bendamustine hydrochloride, one of the drug product, the same drug product now it is available in a liquid injection, which is injection. What you need to do is, as we shown in the left hand side, the top picture, only thing is you have to, with a syringe, you have to take out the solution from the vial and you can give to the patient. Again, some of the disadvantages here I'll tell you. instead of taking 2 ml from the vial, it's a 4 ml vial. You can see 100 mg per 4 ml. Instead of taking 2 ml, you have to take for that particular indication. It's an oncolytic drug, which is used for the cancer treatment. Basically, you just want to give 2 ml of drug product solution, but whereas you might have taken 2.5 ml or 1.5 ml. And you guys knows that minimum effective concentration, therapeutic index, TI, you guys know. If you give the less amount of drug, it is not effective. If you give more amount of drug, it will be toxic to the patient. Okay, this is the again disadvantages here. But the pharma industry was not stopped here. Again, they went back and they had started doing their R&D work. The last one, if you see, can someone please take out this? I don't know how to take. Yes. Now you guys able to see, right? Hello? Yeah. So the pharma industry was not stopped there. Again, what they did, the same liquid injection, they made as a intravenous infusion bag, which is nothing but you might be seeing in regularly day-to-day basis sodium chloride bag, 0.9% saline bags, which typically we'll call it as our 5% dextrose bag or lactated ringers bag. So in which the drug solution was dissolved in a suitable diluent such as 0.9% sodium chloride or whatsoever it is. You have dissolved and keep it ready. So directly you can put a cannula to that infusion bag, like how they will do in a hospital. You can give to the patient. So now you tell the advantages here is nothing, because advantages are more, because you no need to take out from a vial, and you no need to further reconstitute, and where you might have reconstituted with less amount of, you might have added less amount of diluent or more amount of diluent and all. But here the advantage here is it doesn't contain anything. Directly it is ready to use bag. We will call it as a ready to use, which means directly you can infuse this to the patient. So that is the advantage. But disadvantage will be, just imagine other side of the coin. So whenever you are diluting a product much into a lesser concentration, by theory, most of the time there were some scenarios where the higher concentration was less stable, but majoritarily the higher, as the concentration is very high, means towards to the saturation concentration, the drug products will be highly stable. The disadvantages here in the last delivery system was the product may not be stable. You can see the first one may be stable for 2 years or 3 years, whereas the second one may be stable only for a year, whereas the third one will be stable only for 6 months of time. You got it? That's the disadvantage here. Or sometimes you may have to keep that whatever the IV bag which is there at the last, that you may need to refrigerate it, like how the Pfizer vaccine is coming now. It has to be stored at -20, -40, -60 degrees centigrade. Okay, which is not that easy to maintain in a pharmacy stores, and especially in a Indian tropical conditions, where comparatively we have more heat, when you compare with other countries. So that is the disadvantage. Yeah. Coming to another part of evolution process. So there is one more thing. You just want to give into IV form, but whereas your product is when the moment, if your drug product, the moment your drug product started interacting with your infusion solution, which is in the first picture, you can see top there is a powder, down there is a diluent, which is maybe 0.9% sodium chloride or something. So pharmaceutical world, as I said, that it is a very dynamic. So if it is not stable, what you need to do? Separate both the things and put a separator in between both of them, so that until you mix it, both the environments will not be in interactive. Like the powder which is in the top layer won't be in touch with the down layer compartment which is there, the diluent section. Whenever you want to add the diluent, just there will be a separator. The red dotted lines were the separator. It's like your clip. Take out the clip, press forcefully and shake it and give it. So the reason why I'm telling is, during your education also, you guys, always industry will refer students project works and their ideas. Okay, when we started into the industry, we will be in a day-to-day basis activities. We will be always busy and we won't have time to think much about on these innovative ideas. But whereas you guys, youngsters, always will have so much of ideas. When I say that, I don't want to keep both the things, but I want to keep it. Then you gave some people gave idea that to keep a separator in between, then give it to there. So that's the advantage. Then again, the next one you can see, these are bilayer, bilayer pre-filled syringes, in which the top compartment will contain your top compartment will contain your drug diluent. Lower compartment will contain your drug product. You can see here there is a notch. You can see right here there is a notch. What physician or nurse, what they will do, once you want to administer it together, it is not stable. That's why we went to bilayer. Basically, once doctor wants to give, what he will do, he will push little. So once it will come to this notch, once this stopper reaches to this notch, so there is a extra surface where the diluent slowly come into the down compartment. Then you can recon and you can give it. How is the idea? Very good, right? So these are the expectations from youngster, from the academic people. We are expecting, and industry is more keen into it. And this is one idea. There were so many ideas were there, like where you don't want to keep together because of having issues like the product is not stable as together, but separately it is a stable. But you can think about it. Okay. Then again, the another right hand side, you can see this is a vial. This is an injector vial. This is called a pre-filled syringes, whereas this one we will call it as a vial. What you need to do is push it and rotate it. When you push and rotate it through this stopper, whatever the diluent will be there will come into the product solution and will you can use it. But it is one as a ampule to vial, if you went, cost was increased. Yes, we totally agree. But the other side of the story, and very important as a pharmacy like pharmacy fraternity, what we want to understand is, what if the doctor or nurse might have given a wrong dose? It leads to fatal some instances. We know that, right? Therapeutic index and MEC and MIC. When you won't give the exact dose or the desirable dose, it leads to some serious consequences. So that is the advantage here. It is not only about the cost you have to think always. It is about how best you are giving to a patient. How best you are avoiding the complications are involved in it. For example, if you want to give the same vial to a nurse and you say that recon and give it, whereas if you give this pre-filled syringe or this bag or this vial to the nurse, of course they will like these technologies because it doesn't require so much of a laborious procedure. It's a labor work only. Indirectly I can say that it requires so much of effort. They have to take out and they have to add it. It will take so much of time. And that in hospital it is not one patient, they have to dealt with multiple patients. Each and every time to deal with so much of this is not that easy. That's why industry is going towards this evolution. And guys, whenever you are doing project work, always not only about the traditional technologies, think about these aspects also. You can give your thought process so that industry is there always, to take into the next level. They will help you out whether that is sustainable idea or not, or is there in advantages are there, are always pharmaceutical industry. We all know that risk to benefit ratio, it will work. If the benefit is more than the risk, we will take that as a approach and we'll go for it. Like corona COVID vaccine, no vaccine was not approved, but industry is selling because it is having comparatively benefits than the risk. So because all the studies was not done. So put your thought process in these sort of technologies and give us some ideas and publish the articles. Always don't think that nobody is watching. Industry is very keen on that your your literature research articles. Industry will always watch your whatever you are publishing or whatever you are doing, and we will always look into those things, and we will work on it whether it's a sustainable idea or we cannot go ahead. Okay, then is the flow is fine, or I can go a little faster, or I can go a little slower? Please give your feedback, so accordingly I can go. Can someone respond, please? The flow is fine. Are you able to, are you guys able to understand what I am saying? Or I need to go a little slower, or I can go a little faster, or the same pace is fine? Please, somebody can tell me, please.
Same pace is fine, sir. We are
Yeah,
able to understand it.
Yeah, thank you. Okay, then this is one case study. The reason why I kept 0.9% sodium chloride and 5% dextrose, you might seen this very regularly, right? This is saline with dextrose combination infusion bag. You might be seen in a pharmaceutical nursing stations very commonly. Consider you want to develop this product, 5% dextrose and 0.9% sodium chloride injection, you want to make it. So it is all about, when it comes to the industry, if you want to make this drug product, what you need to do, those aspects I'm going to tell to you guys now. Okay, first you have to see what API. Consider here dextrose and sodium chloride we will consider as a API, because these were actives in this IV bag. Okay, in which dextrose we are using and sodium chloride you are using. Okay, so first you have to select who is supplying that particular APIs. Dextrose who is supplying, and sodium chloride who is supplying. The pharmaceutical grade. You cannot say that sodium chloride, I can take a salt and I can make this. Okay, don't do that. You have to use always parenteral grade APIs or excipients. The reason why I'm saying parenteral grade, it will have low amount of spores, endotoxins free or very low endotoxins. What happens if endotoxins are there in your formulation? Your body will consider that as a foreign body. You'll get a fever. Again, same, whatever you had read in B-Pharm pharmacology. When you inject to rabbit, any foreign body, what happens? The rabbit's body temperature will go high. Immediately, immune system will start responding. All these things will happen. So, selection of API and excipients is the first thing and essential and foremost thing which you need to do. And upcoming slides, I'm going to tell how to select those also. And there were so many vendors are there in the market, in the pharmaceutical world, who will supply these dextrose and sodium chloride, and they will supply you, and you can purchase from them, and they will sell with a certificate. Okay, always ensure you are getting with a certificate. In a pharmaceutical terms, we will call that certificate as certificate of analysis, or C of A, or somebody will call as COA, certificate of analysis, which is nothing but before sending to you that excipient, they already did the they already analyzed that, and they had made a certificate and they are saying it, this is the purity. I'm saying like you are expecting 100% purity of sodium chloride, then they will say that their product is 99% stable or 90% stable, or some will be, for example, if you use homemade Tata salt or something, it may not be 100%, it may be 95%, whereas other 5% will be some severe impurities might be there. So always very important during your project work also, whenever you are taking any APA or excipient, always ask certificate of analysis. Like whenever you do anything, right, you will ask certificate. It's like a warranty certificate, like that you need to ask. Then so the top picture you can see, this is a typical manufacturing vessel, in a pharmaceutical industries, which we basically we will use in lab scale, like in B Farm or M Farm, we may use glass beakers, 500 ml, 100 ml, or depend on how much ml you want to make it. 10 ml or 100 ml, 1 liter, 2 liter beakers you may use. But in pharmaceutical world, we will use these sort of vessels where it will have additional provisions. Like for example, suddenly you want to give nitrogen environment to that. In a lab scale batch, if you want to give to into a beaker, what you have to do, you have to take one tube, pneumatic tubes you have to take, inert pneumatic tubes, then you have to push it. Whereas these vessels will have separate provisions, it will have spargers, other provisions to give other gases inside to this manufacturing tanks. We will call these are manufacturing vessels or manufacturing tanks. Again, as we all read in B Farm, everything is unit operations. I think in B Farm we all will do that 0.9% sodium chloride preparations. You guys all done that 0.9% sodium chloride preparation in your B Farm, right? Can anybody respond, please? You did or you didn't do? Okay, I'll consider that as a yes. Okay, if you guys are not responding, I'll consider that as a yes. So
No, sir.
yeah. So what you did basically, 0.9% sodium chloride means 9 mg per ml of sodium chloride you need to take it, and you have to dissolve in a water for injection or sterile water for injection. Then you have to mix it. Then you have to do the autoclaving. So these are the steps involved in it. So in laboratory grade, whatever the chemicals which we use, or those were different than in a pharmaceutical world, what you guys, when it comes to the industries. Here we will use very high pure grade materials we will use. Then again, when you start your project, these are the studies which you need to do whenever you start a project. Consider the same 0.9% sodium chloride, 5% dextrose you want to make it. So what you need to do? First, you have to select down, you can see propellers and impellers, different types of agitators are there, you can see, which typically we will use in a pharmaceutical field. High shear mixtures, high shear impellers, high shear stirrer rods, which we will use. Agitator depends on. For example, if you are using a regular 0.9% sodium chloride, and which is very soluble, easily soluble in your water for injection, you no need to use high speed or more advanced stirrer rods. You can use a simple vortex blades, you can use agitators, you can use, and you can mix it. If you use a high grade one, that is okay. Comparatively, you are reducing the time of manufacturing. For example, if you use a regular blade, this propeller or impeller, if you use, it will take 10 minutes to dissolve. But whereas if you take this, the last one, high shear, if you use this one, it may go within a 2 minutes or minute also sometimes. So that is the capacity. Okay, so that is called stirring rate and stirring speeds. Again, whenever you start, like you might have collected into a you might have collected a beaker, then you might have then you added a little amount of water into that, then you might have added the sodium chloride, and you started rotating it with a magnetic stirrer by using beads or overhead stirrer with the propellers or impellers or whatever it is. Then how much speed I need to keep it there. Because when you go with here, manufacturing tank, nothing is visible. You can see only digital displays there. How much speed you have to keep it, you can see only. Okay. It is not like, where you can slowly increase or decrease, that and all is not possible there. So you have, whenever you are doing a developmental at R&D, you have to check how much stirring speed I need to keep, how much time I need to keep, I need to stir by using this simple R to get a clear solution. So these are very important. Okay. Then order of addition. Consider you had taken, you might have prepared 0.9% sodium chloride where you have only one active, which is sodium chloride. Consider the example which the reason wantedly I kept was 5% dextrose and 0.9% sodium chloride. So which one to add first? Or do I can add both together? In B pharmacy, we will do this order of addition studies. Whenever we start making any emulsion or anything, suspension or some dosage forms or ointments, whenever you are doing, you need to have a flow. First you need to have one excipient followed by another excipient followed by another active ingredients. So that is called in a pharmaceutical terminology order of addition studies. Means which to added first. For example, do I can add first sodium chloride, or I can add dextrose, or together also I can add. So what you need to do, you have to make different experiment, different combination of experiment by adding first dextrose followed by sodium chloride. Then next experiment, first sodium chloride followed by dextrose. And the next experiment, add both together and do it. Then keep stirring rate and stirring speed as a constant. Then you will check it out how much time it is taking, or is there an issue. For example, what happens if you didn't follow while making a emulsion? Just imagine phase separation will happen. That's all. The same thing will happen here if you are not following the order of addition properly, or if you are not doing a R&D here, how to add and which to add first. Then your impurities or whatever the degradants which may go very high. So that is very important. You have to establish order of addition studies. Then compatibility studies, which is nothing but, as I said, everything is a unit operation. Consider here you had added, you prepared in a beaker you had prepared your saline solution in your B form. Then that saline solution you might have filtered with filter paper, right? You might have taken a glass funnel or plastic funnel, and another beaker, clean beaker you might have taken and you might have filtered it. But whereas here in the industry, it is not possible, like, because you are handling with thousands of liters. So you cannot do, taking a funnel or something. So here what will happen is some tubings we will use, which was mentioned here, different sizes or different grades of tubings which we will use. Then different filters we will use. These are called as filter capsules, and filter disc filters also we will use, which is nothing but your regular filter papers. We will connect these. This is called a manufacturing area. Then the next room will be considered as a filtration area. The next room will be considered as a filling area. Next one will be considered as a sterilization area. Next area will be considered as a labeling area. You got it now? Everything is a unit operation. Each area will have its own unique importance. So to move the solution from one area to another area, we will use some, adjoints, like, these sort of tubings we will use it. You can see here, this vessel will have down a tap. We will call in a pharmaceutical world that as a ports, PORT, ports we will call it as. That to that ports we will connect these tubings, and the next vessel will be connected with a, consider this is your bulk solution tank, and here this is a filling tank. Here you might have kept your filter. Both the filters, filter out and filter in was connected with a tubing, and once you start pressurizing here it, okay, generally whatever you do in your B form or M form, that's a gravity filtration. You'll pour on top it, and through a gravity it will go it. In industry, we won't use gravity filtration because it will take so much time. What we will do here, we will use a pressure or force filtration, which is nothing but we will apply a air on top of it, air or nitrogen on top of it, which is nothing but it will forcefully push the solution from this tank to this tank through these tubings with the filter, then it will come out. Okay, or we will apply sometimes vacuum filtration also, which is nothing but we will keep a vacuum pump here, and then we will started sucking it out, then the solution will come. So there is a echo. Somebody please mute yourself. Once if you have a question, please unmute yourself and ask. So the reason why we won't go for the gravity filtration in pharmaceutical industry, the volumes are very high, we cannot do it, and it has to be happen very fast. Okay, that's why we'll use this pressure filtration or vacuum filtration. Okay, then I told you why we have to use this tubing and why we need to use these filters. Then again, you have to do the compatibility studies with different things like, you know, stainless steel, glass, Teflon, filters, tubings, gasket, and your material of construction. Consider there were some drug products. Okay, drug product is nothing but, you know, which you are seeing outside, you know, formulation are nothing but, consider this 5% dextrose and 0.9% sodium chloride is a drug product. So sometimes these excipients or these APIs may not be compatible with this stainless steel impellers or stainless steel vessels which you are using, which means it may corrosion. For example, some of the products manufacturing, we will use concentrated hydrochloric acid. What happens when you add directly concentrated hydrochloric acid to a stainless steel vessel? It will start corroding, right? And again, the next story, you know, what happens if you give the corrosion to a patient, a corrosive rust to a patient? It's a fatal, like, you know, he will die, and some best cases it will land up into a hospital, you know, he will land up into a hospital. The worst case is he may die also. So in such cases, what you need to do is you have to establish the compatibility. So sometimes these SS vessels which you are using for the manufacturing may not be compatible. So in such cases, you have to use the glass vessels. Okay, typically glass lined vessels will be there in a pharmaceutical industry, and sometimes glass also may be not suitable. Consider whenever you are using the highly basic formulation, the glass is not stable. If your pH is above 9 or 10, which is highly basic, then the glass also will, you know, literally you can see the glass flakes, you know, the glass flakes you can see into your solution. Okay, what happens when the glass flakes into a IV set? You know, again, it will have a serious consequences. So in such cases, you are land up with SS is not compatible. SS is nothing but stainless steel, and glass also not compatible. Then again, there were some one more material of construction is called Teflon. Teflon, you know, regularly you might be seeing whenever, you know, plastic pipes are, when they are connecting, they'll use this Teflon tapes. The same material, bigger manufacturing tanks will be there, in which the manufacturing tank is made up of entirely with a Teflon, which is a inert environment. It won't interact at all. But if you want to use Teflon vessels, it is very costly. It requires separate manufacturing vessels because all the manufacturing facilities does not have the Teflon vessels. Okay, then again, there will be so many tubings are there, and which tube you need to use when you start your project, you don't know, and even I also don't know. Then what you need to do is you have to take all these different tubings, then you have to check the compatibility, which tubing your data is fine. Then same with the filters also, which filter you need to use. Then again, consider majority cases, the pharmaceutical drug products are stable at certain pHs. Okay, that's why we have to do the pH suitability studies, which are nothing but what happens if your product pH goes beyond. For example, this 5% sodium chloride and 0.9% sodium chloride pH is 4.5 to 6.5. What happens beyond 6.5 and less than 4.5? Okay, being at research and development, you have to understand what happens if it goes beyond. Okay, those are the studies which you need to understand. The reason why is tomorrow, if at all in a manufacturing facility, you got a call from manufacturer saying that whatever R&D which you had done, we are getting some impurities. What happened? They will ask you. Then during R&D, you should do this pH compatibility studies, and you have to see what might go wrong when you use the alternate pHs. Okay, that is the importance here. Then again, effect of dissolved oxygen and headspace oxygen, which is nothing but generally what happens when nothing is there in your atmosphere contains mixture of nitrogen and oxygen and carbon dioxide, right? Majoritarily. Consider in lab environment, it will be less of carbon dioxide, more of oxygen and nitrogen. Okay, nitrogen consider only 10 to 12% is there. You consider in a product. Whether the 10 to 10% is fine for your drug product is not, we have to understand. Which means if your product contain oxidative impurities, which is nothing but if your product was interacted with oxygen, your product will form some degradants which are oxidative degradants. So in such cases, what you need to give? You need to give a inert environment, either dissolved oxygen or headspace oxygen. So in such cases, either nitrogen, argon, these are the inert gases. So these gases you need to give to the manufacturing vessel while making it. Okay, that you need to establish. Then photostability studies, for example, photostability is nothing but generally our pharmaceutical products will be exposed to a light, right? What extent this product can be exposed to light? For example, if your product shelf life is 2 years, 2 years the product is stable. 2 years of time, how much light it can be exposed, and what if the light, if like for example, some will be there where light degradants, photolytic degradants we'll call it as. If it exposed to the light, it will start degrading out. So in such cases, what we will use? In B Farm, what we did? Okay, we used to take amber vials, right? Amber beakers or amber, while making the syrups and all, we will take amber bottles, right? That is why we take the product may not be stable in a clear vial or beaker. So that's why in such cases we will take the amber. Amber, if you use, what happens is amber will inhibit the light, not completely, but it will inhibit the light. So that's the advantage. So for that, you need to do the photostability studies by selecting different packings. Like, if at all you purchase the ampicillin vial, it won't be like directly, they won't give a vial, right? If at all ampicillin you want to purchase from a outside, it won't be in a vial, right? They might have kept in a carton. Inside the carton, the vial might be there, right? So those are the studies you need to establish. But some injections like 0.9% saline bags and all will be there directly, they'll give you. But whereas some vials, they won't give you directly. They'll keep inside a carton. Carton is nothing but, like a Amazon or Flipkart box. Whenever you order any product, it will be inside a box, right? Like that, in a box, your drug product will come. So you need to establish whether that is required or not. How to establish that? You need to run the photostability studies at your R&D. Next, thermal cycling studies. Thermal cyclings are nothing but transportation feasibility purpose. For example, if the product is moving from USA to India, consider COVID vaccine only moving from United States to India. Indian temperatures are different and USA temperatures are different, right? So what happens whenever your drug product exposed to higher temperature to lower temperature or lower temperature to higher temperature, what happens? You need to establish. right? Today morning, you can see Pfizer has sent a data saying that the drug product is also COVID vaccine, Pfizer vaccine also stable in refrigerated at 1 month. Okay. How they might have come up with that? They might have done these thermal cycling studies. Okay. Like some of the real scenarios I am connecting out. Okay. Whenever you are doing any product, you need to do the product stability at different temperatures. Okay. Then whole time studies are nothing but how much time you can hold this product. For example, in B Farm, whenever you are making this saline, you might have manufactured the saline, then you might have went home. The next day you might have came, and next day you might have taken, and you might have filled into a bottle or vial, and you might have submitted to your lecturer for for your review purpose. But industry is not like that. You have to establish how far the product is stable. Some products are stable only in that vessel only for hours. Some products are stable in minutes also. And in that case, what you need to do is, if the product is not stable, what is your immediate thought process? You can give a refrigerated environment. In the sense, you put some ice cubes over there. As we know by theory, as the temperature lowers, your product degradation will be lower. Okay, those are the thought process you should keep always. If your product is not stable at that particular environment, immediately you have to think how to do it. Okay, then effect of excipient concentration. Again, you should not add excipients just like that, or consider 0.9% sodium chloride, 9 mg per ml will be there. You should not add 10 mg per ml or 80 mg per ml. You should use always only 9 mg per ml. But there is little difference can be done, but you should be very careful on that part. Then vials and rubber stoppers, compatibility studies. There were so many stoppers which I had shown here. There were so many stoppers from so many manufacturing vendors were there. So many vials were there. Which vial or which stopper you need to select? You have to do the again R&D compatibility studies. Which stopper or which vial you need to use. Okay. Then extractable leachable studies, which are nothing but like when in the product in contact with the drug product solution, what is coming out from the drug drug product or particular that bag, you need to establish. Those are called extractable and leachable. Withdrawable volume, which is nothing but consider this 5% dextrose bag. You can see here they had mentioned 500 ml. Okay, which is nothing but if nurse or physician want to take out, at least they should take out the 500 ml from that. So for that, there is a guidance in the pharmaceutical industry. USP, like United States Pharmacopoeia, will say how much ml you need to fill, and we need to do some withdrawable volume studies by doing. For example, if you want to take out 500 ml from this bag, if you keep exactly 500 ml, you cannot take 500 ml because there will be always some dead volume will be there, where you can see in these regions some some drug product solution which you cannot take out. Those are called dead volumes which you cannot take out. So for that, you need to add some extra volume inside that, so that the physician or nurse can take out the exact 500 ml. Okay, those are called the withdrawable volume studies. Okay, so you need to very careful on that. And again, it was mentioned here as 500 ml, and you cannot say that 450 ml or 600 ml or 1 liter you can fill it, right? You should keep there is a guidance in the pharmaceutical industry. Exactly, you need to keep how much ml is enough to take out. Okay, then I'll show you a video how the sterile injectable was basically made. You can see here. Everybody able to see screen, right?
Yes, sir.
Yeah.
Yes, sir.
Yeah. So this is not in the R&D level. This is in the manufacturing, like manufacturing level, how it will be. Okay, these are called isolators, which is nothing but you won't be exposed to the product. Everything will happen inside only. Okay, everything will be inside only, so that there is no point of sterile breach or something. Okay, these are called isolators. Okay, if at all you want to like everything on its own, it won't happen, right? So everything won't happen on its own, right? Won't happen, right? So you need to use these gloves to put your hands inside, then do the process. Whatever mixing you want to do, or whatever stoppering you want to do, everything you need to do, use these glove boxes. So this is the very advanced and current manufacturing standard, and this is called how many of you guys went to IPC? If you didn't go, please at least your BPharm or MPharm time, at least once you go to IPC or at least to a pharmaceutical company project work you go, so that you will understand all these things more familiarly. You will see directly. When you go to IPC, these vendors will bring all these things so that you can get it there. So this is called turntable, where the vials will be kept there. You can see everything is automatic. How vials were taken here. Okay, once the vial is taken, you can see here how the vials are washing inside from outside. You can see here different position. Here the position is around the degrees of exposure will be different, so that inside and outside the vial will be washed. Okay, you can see here. Once the vials got washed, then again it will come to the conveyor, which typically you will call in pharmaceutical industry. And I just want to show you, this is called Teflon. This material is called Teflon, highly resistant, and it won't interact with anything, and highly stable. So these are the Teflon. Okay, so again, as I said, everything is an automatic process. No need of manual intervention majoritarily. Only thing is, everything you'll have a display, you need to operate through that. You can see here again the vials are passing through a tunnel, because vials you should not use directly. It has to be sterilized. For example, like in your BPharm or MPharm microbiology, before taking your vessels, you will heat before you take it. What happens is so everybody able to see screen, right? You can see here so here you can see this is filling. This is another unit manufacturing. You can see here these are the filling needles. How these filling needles was connected with a tubing. So you got it now, what I told in the first slide. Everything is connected with one to another. So that's why consider this is not compatible with the product solution. What happens? Your product is not stable again. Okay, that's why we need to do all these studies before you are giving to the manufacturer. Okay, so this is called dosage, which is nothing but consider when our professor says that you fill the bottle with the 550 ml. How you do it? You will take a measuring cylinder or volumetric flask and you will measure it and you will transfer it. But whereas in the manufacturing facility, you cannot do, because here lakhs of numbers of vials will be filled, right? Per a second, 1,000 vials will be filled. So in such cases, what you need to do is you will have a load cell, which is nothing but beneath this vial, there will be a balances weighing box. weighing will be there. Those are called load cells. Okay. Somebody please mute yourself. There was so much of echo. Yeah, thank you. So once you fill, immediately it will come to this area where the wire weight will be calculated. How the weight will be calculated? Density is equal to mass by value. For example, you will measure the density in your R&D, then you will check it out how much mass should be there. Then here there is a balance, and it will give, yeah, this is your load cell. As I said, here the weights will come automatically, and here you will give how much ml you need to fill everything, and what is the lower fill weight, higher fill weight you need to give. And I said, right, 500 ml if you want to manufacture, there will be some, we have some guidances. So according to the guidances, we will fill what is the target ml. Target ml is 10 ml, but less higher you can fill 10.5, lower you can fill 9.5 ml. What happens if it goes beyond or above? It will be rejected on the line only. It won't go. Okay. And these are the load cell. 4 load cell will be there. Nothing but 4 balances will be there. All these 4 vials weight will be taken, and it will go to the next step. If it passes, then only it will go, or else the system automatically will reject. Okay. Here, whatever the demo which I am showing here, it is not 100% weighing it is doing. Only few vials only it is taking. You can see here. Since it is between 10.5 and 9.5, it said okay. If it is beyond that, it says not okay, and it will reject directly. Okay. Yeah. So nowadays these lines were 100% visual, in like 100% weighs we will take, not even one vial also we won't omit it. But here some old techniques, like every half an hour they'll take few vials, and these are called rubber stoppers. You can see here also again tubings were there to suck the stoppers. This is called stopper bowel. So where in a sterile environment you will pour all of your rubber stopper, then this is how the stoppering will be done. Okay, then again you are like how we do in B farm. If you want to arrange the vials, you will keep side by side, right? In industry, this is called hexagonal packing system we will consider. Okay, how it will automatically pack it. You can see now how the hexagonal pattern will be. Okay, this is called hexagonal. It won't be next to next. 2 vials will be there. In between these 2 vials, the next vial will be there, so that the vials will be in a hexagonal pattern. Okay, like that it will be. And then again stoppering will be done. Again it will come out. Then this is the sealing area, where first these are the some of the sensors which industry will use. Consider if your vial doesn't contain the stopper, there itself it will reject. It happens sometimes. It is no process is 100% absolute. Always process will have its own defects. So that's why we will put always in the industry, we will put always some sensors there. It will sense automatically. If the stopper is not there, it will reject it. Then here you can see it is crimping it. Crimping is crimping was done, which is nothing but with a sealing is done. Then once after was sealing, again it will come. Again external vial washing will happen, which is nothing but, since it's these are pharmaceutical preparations. For example, you might have there might be some spillage to the on top of the vial. For example, while loading, glass breakage is very common phenomena, right? Due to friction between vial to vial, or when it is going in a conveyor belt, some vials will get broken. Whatever the broken vial solution will come out, right? That solution will be will be touched with this vials. That is very dangerous, right? So that's the reason vial washing also will happen at the end. So you can see here there were some nozzles where water will be sprayed on top of it. Then it will go to the labeling part. So okay, so so until this time, you guys, heard what R&D is and how R&D things will work. Everything is fine, or somebody having doubts, please let me know. The coming slides will be more of, in the pharmaceutical industry, once you come, what you need to do those things. That's why. Okay, I'll consider that as a yes again. Yeah, again, once you come to the R&D, they'll use some of the terms, 505J, 505B1, B2, B3, B4, so many things they'll start talking it. 505J is nothing but in already there, saline, whatever the 5% extra 0.9% saline already there in the market, right? You are making your own 5% and 0.9% sodium chloride, which is called as a generic version of the product. This is called as a 505J. 505B2 nothing but, consider earlier guy might have kept in a plastic bag, but you made it in a pre-filled syringes, or you made a little difference based on the clinical data. For example, clinical data is saying that 0.9% sodium chloride and 5% dextrose combination is not a good ideal diluent, where they might have, clinical team might have said to you that 0.45% and 2.5% dextrose combination is good. Then if you are doing such case, then you will consider as a 505B2. Again, different patent terminologies they'll use. Basically 4 types of paragraphs will be there. Para 1, 2, 3, 4. One is nothing but very old drugs. Consider your paracetamol drug. Okay, very old drug where there is no patents at all. Para 2 is nothing but there were patents, but patents got expired. Para 3 is nothing but patents were active, which is going to expire in coming year or 2 years of time, which is going to expire. Because to do the R&D, you require some time, right? More than 2 years it will take once you start doing your R&D and before you get approval and launch your product into the market. Then para 4 is nothing but you want to non-infringing way. For example, he might have done with a one process, but you want to do with another process. Is nothing but, for example, he used a process to manufacture the drug product, but you want to use a B process to make the same drug product, or then which is nothing but in a non-infringed way. Like whenever any patent is there, you cannot manufacture the same product till 20 years of time. So that's the Patent Protection Act. So if you want to do the out of the patent, then you have to approach the para 4 approach, which is nothing but out of patent approach you need to do it. Then generally, this is not standard, but company to company it will vary. What we will do, we will prepare stage gates. Nothing but, for your B Pharm, M Pharm projects, you will do first literature collection, followed by materials collection, followed by excipient analysis, then summarizing results. All these different steps will be there, right? In the same way, in R&D also we will have different process. Like for example, first what we will do, we will summarize existing information. For example, 0.9%, 5% dextrose product only manufacturing. Then I'll see what it contains, who are there already in the market, and what patents are there, how people are manufacturing that, and all these information we will keep it. Okay, then we will make a report out of it. Okay, then in the stage 2, what we will do, R&D development, which we had seen in the slide 5. All these studies we will do. Then we will go for the exhibit batches, which is nothing but whatever you have developed that drug product, you will transfer into this manufacturing facility. Okay, that is called exhibit batches. Okay, then next you will go for once after exhibit batches, you will file that information with the regulatory agency, like which country you want to get the approval. For example, USA or Europe or whatsoever it is, in India, whatsoever it is. Then once after you get approval from that agency, then you will go for the commercialization, which is nothing but you will start selling the product into the patient, like selling into the market. Exhibit batches are not the commercial batches. Okay, only commercial batches only. These are exhibit batches are nothing but for the filing purpose or to generate the data. Because once you, whatever you do in R&D, whatever you do, we might have done in the B Farm and M Farm lab, and it is different to the R&D, and it is very different to the manufacturing site. So that's why it is very essential that exhibit batches you need to do. Okay, stage gate preparations, you have to select so much of data, you need to collect it, and you have to make a proposal. You know what you want to make it. You want to make a generic product, or you want to make a RLD product, or this will come from your management, by the way. But you need to understand what you want to make. You want to make a novel new product, or you want to make a generic product. Okay, all these you have to make in the proposal. Then generally what you need to do, then API excipients you need to select, and grades you need to select, and packaging components, nothing but vials, stopper, seals, you need to select. And then you need to understand, is there any patents are there? Then you need to do the risk assessment. Then you have to get the test license and import license from applications from the CDSCO. Then manufacturing process you need to make it. Then batch sizes you need to select it, and commercial volumes. For example, whenever you do a mini product, you need to get the data from the commercial teams, how much requirement in the market for that particular drug product. Then accordingly you will fix is the batch sizes and commercial values. Then again, this is one, like at this time it is not important, but you just make a note of it. Drug master file, DMF, which is nothing but only you have to use most of the times DMF grade materials, and different types of DMFs will be there. DMFs will be given for the manufacturing site facilities and drug product substances, packaging materials, excipients, and accepted information. Okay, then in the previous slide I said, right, some of the guidelines which we will follow, which are nothing but ICH, International Conference on Harmonization, which you guys might be read in BFarm, both MFarm as well. It will give some guidance to the industry in respect to the quality, safety, efficacy, and multidisciplinary guidelines. And generally these guidelines are applicable only to the European EU and Japan and USA, but it is not like that. Every country is accepting this ICH guidance because these are most stringent guidelines. So that's why. Then so far I gave rough idea how the industry will be, and what you need to, and what to do, and how industry is progressing, and how the manufacturing facility will be before transferring your project to exhibit batches. What and all studies you need to do in a R&D scale, I had told you. Then these are the some of the disciplines which you have to have whenever you come to the industry, whenever you come to the manufacturing industry, or whenever you work in a pharmacovigilance or whatsoever it is. Once you become a pharmacist or pharmacist student or pharmaceutical fraternity you come, you should have some integrity. Okay, the reason the integrity, why it is required, I'll tell you. As a person you should have integrity, and we all pharmacy oath we have taken. Okay, so you have your own personal integrity, and when you come to the industry you should follow some data integrity, which is nothing but for example, in BFarm, MFarm, majority of the people will do, they will manipulate their data. If the dissolution is not coming properly and your observance is not coming properly, they will manipulate the values and they will do it. But in industry you should not do it, because at the end the drug they might have administered to you or your family members or your loved one, which is nothing but your son or daughter, or your parents might have got the same drug product. Okay, you got it what I mean? That's the seriousness here. So whatever you are doing, once after you come to the industries, you should have an integrity. And if you are not having integrity, then one or other day you will get caught. Once you get caught, no company is going to take you inside. Okay, that's the breach of trust, not only between you and your colleagues and your management, it is between the regulatory agencies. So generally what happens in a data life cycle, any data integrity, what is a data? whatever you do in your B Form project or M Form project or any one lab experiment, whatever you say that today you might have prepared a simple syrup solution, 66.7% syrup you made. In a process, you did series of steps, right? You had taken syrup and you had added and you diluted and you had mixed it and you had checked it quality and you had documented in your laboratory notebook and you have checked with your professor. All these things you have to document it. In industry, if you are not documented, and we will consider that as you never did that experiment, you never did that. So always documentation is very, very important and essential. So data, you once the created data, processing, review, retention and destructions, these are the life cycle of any data. You should follow guidelines, and your wherever company you are, always you have to have some ethics, because if you manipulated anything, that product might come to your patient, you only. So imagine your 0.9% sodium chloride, whatever you had prepared, which might have injected, a nurse is injecting to you, and you know that you made a mistake there, and there is a sterility breach in that particular day, and you haven't added but exact amount, and you haven't checked the pH properly. Just imagine how you are going to be. Will you accept such drugs when you are injecting to you? Don't know, right? So the same you should. Okay, always you should think about the patient. The patient, end patient might be your family members or you maybe. So think about it, and your friends. So you should have integrity. You should never commit any fraudulent activities, and you should never manipulate any data. Simple thing. If you did some some error came, consider you are preparing 0.9% sodium chloride, and your pH should be 4.5 to 5.5, and you got 4. Dump it, or you speak to your professor. Professor will tell you how to avoid the situation. Okay, you do with some pH adjustment, you do adjustment, pH adjust, then you go for the sterilization and the packing. But before that, sometimes you cannot do the pH adjustment. Some products are not subject of the pH adjustment. That time you just dump the project, that particular batch, it is fine. But you just imagine you circulated this product into the industry, and once it went to the patient, you might have got a feedback from the industry that one patient was died, or one patient has received adverse events after injecting this saline. Then what you need to do? Not only you need to recall this your alone lot, you have to halt your entire 0.9% sodium chloride batches, whatever you manufactured so far, and everything will go for a scrutiny. Everything they will check it out, and quality will check it out, and agency will come to you, and you might be regularly seeing in a pharmaceutical fraternity, Dr. Reddy's or Pfizer might have observations from the regulatory inspectors. FDA inspected their facility, they gave observation. These are the common things which you might be hearing regularly in the newspaper. So these things will come. What happens once if FDA or anybody will inspect your site, and you are not following the practices? It's a matter of trust. Once you lose the trust, FDA or regulatory agencies, nobody will trust you. Moving forward, whatever, not only for the 0.9% sodium chloride, whatever you manufactured in that facility, everything will go for the scrutiny. What happens in that time? You wasted company's reputation because of you. Because of your mistake, company's reputation gone damaged, and you are not selling your product into the market. What happened when you are not selling into the product manufacturing? Company is in loss, right? When they are not selling, what happens? You may lose your job, and ultimately, all these consequences will be there. So you should have always your integrity. Whenever you do a mistake, immediately tell to your manager. He will tell you whether to go ahead or dump it. Go ahead and do it. When you do these things in industry, your managers will appreciate it. When you say mistakes, manager will appreciate. If your manager will have an answer for, there will be some process where you can do little more. For example, You are doing a filtration, suddenly your filter paper got damaged. Your filtration, like in a lab, in B Farm, M Farm, when you are filtering, when you filter paper got turned off, you will do like that only. But industry, it is not like that. You can do one more time filtration. It's a matter of taking one more filter and do it. It's a matter of hour or 2, 3 hours maximum it will take. But if you are not filtering, what happens? You are sending fibers, dust particles, everything into your formulation. What happens if that dust particles, everything comes into the product? The patient will die. That's all. It will obstruct your veins, and it will stop a blackhead, and your heart will stop. So these are the consequences, and you may be the consumer one or other day. So keep it in mind. Always you have some ethics when you come to the industries. Please have ethics. You took a oath in a pharmacy. As your professors always says that, pharmacy oath and how it is very important. In industry also, it is very important. You should always have values. Then again, industry, for the data integrity, we will follow some principles. It is called ALCOA plus, which is nothing but ALCOA plus is nothing but complete, consistent and enduring. Whatever you do, attributable, which is nothing but whatever you do, that should be identified by someone. For example, a person today did something. In pharmaceutical industry, it is not like that one guy only will do everything. You may do some part of experiment, and some part of experiment will be done by someone else, right? Whatever the A person will be done, the B person should understand whatever he did, and he should understand it. Okay, so that is nothing but called attributable. Legible, which is nothing but whatever you document it, it should be in a readable format, in a permanent format, and it should be accessible to anyone. Then contemporaneous, which is nothing but whenever you are doing the experiment, you should do in online. Like, in a B Farm and M Farm, some students will do before submitting the records, they will do the writing the experiment. You should not do that. You should do in a contemporaneous manner. Once you start, you had taken beaker, right? I had taken a beaker. Then I had taken, then I had taken a measuring cylinder. Yes, taken a measuring cylinder. Taken 50 ml, measured 50 ml of water, measured 50 ml of water. You start recording. Online you need to do. It is called contemporaneous. Consider tomorrow you come to the industry. In B Farm, it may be 2-3 steps will be there. In industry, as I shown, it's a complex way. It will have thousands of steps will be there, which you cannot read. Do you remember what you had day before yesterday night, your dinner, lunch? No, right? That's all. So that's why this is very important, that you should write whatever you do in the process. That is called contemporaneous. Original, the first recording, true copy. Always, record the originals. Don't manipulate the data here. And accurate. Always keep the accurate information, truth information. Okay, which we had taken a oath. The pharmacist, we had taken a oath. You have to keep the accurate information. What happens if you are not following any of this principle? Patients will die. Then again, this data integrity extended to plus, which is nothing but complete, consistent and enduring, which is nothing but metadata. Sometimes whatever observations books will be there, like in your B Farm, from that observations books you will transcribe into your record, right? That observation books you should not destroy. You should keep it. That is called metadata. Okay, then consistent, which like you should follow always good documentation practices, which is nothing but you should not rub it off, and you should not wipe, you should not use whitener, or you should not erase it like anything. If at all you did any mistake, put a single strike. That's all. Then on top, if you write it, and then you write there information was added wrongly, then corrected, then you write it, sign, make a sign. That's all. Every process having a remedy. Remedy. Always speak with your professors whenever you are doing any mistakes in the lab, or when you comes to the industries, you speak with your managers or your seniors, they will tell you how to do it. When you do it, you also don't want to do a mistake. It happened. It is your responsibility to tell to your professors or to your managers in industry, yes, I did a mistake, how to come out of this situation. Then they will tell you how to come out of it. It requires sometimes some revenue losses. You may have to dump it, but it is the right thing to do it. Somebody please mute yourself if you are not in, if you have any doubt, please, or else you mute yourself. Okay. So this is one of the example which I gave. This is the warning letters. You might be reading regularly. Pharmaceutical companies are getting warning letters. Majority warning letters, 53% was related to the USA, then 24% to the India. And this is the pictorial, and it was in a public domain. It was there, and you can see here majority of the warning letters related to the data integrity, which is nothing but they falsified or they manipulated the data. Okay, what happens when you manipulated the data? Patients will die, and you lose the trust with your company as your next colleague. For example, if my colleague is doing a manipulation, if I came to know one day, then I will never believe him. And it is your responsibility when you comes to the industry or during your B Farm, M Farm, when your next one is doing a mistake, it is your responsibility. Since you had taken a pharmacy oath, you have to tell to your professor or lecturer or to your in industry, to your manager immediately that he is doing a falsification of data. Okay, that's the integrity you should have. Then we came to a discussion session. Thank you so much for giving me the opportunity, and we have 15 minutes of time. Professors, whoever in the call, and students, whatever the doubts you have, you can give a feedback to me, and you can tell to me, you can wherever you have little more explanation or whatever it is, you can tell to me. But one thing is, you should have your integrity. Even in a B Farm or M Farm also, if you might have did so far mistakes, that's okay. Yes, it's a right time. You are a pharmacy student. You took a oath that you won't do the manipulation. Moving forward, you do a right thing. Only thing is what your professor will do. He will give a warning to you, and he will encourage you, because you acknowledge your mistake. And if you say that, yeah, I did a mistake, sir, and I'll do tomorrow, I'll repeat that experiment. If you say, he will be very happy. I'm saying you, I'll be very happy if you say in an industry as well. And I did it in my B Farm. Whenever I had a problem, when my emulsion got broken, or when I'm in a cognitive, when I'm unable to dissect or unable to take out, I always told to my lecturers, sir, I didn't make it. Can you help me out? I'll do tomorrow. I need some more time. I need some more time to make it perfect. They are encouraged me. And in fact, your lecturers and your professors and your principals, your management also will do the same. Be open and have that culture, integrity culture. Whatever the culture you imbibe in your studies, the same will come to your professional environment when you come to the pharmaceutical industry. So that is very important. Learning things today or tomorrow you will learn, but your character and your personal integrity is matters there. Excuse me. And it's time for the discussion. Please, if you have any questions or you want to give the feedback, open, and your principals are and coordinators and lecturers, whoever part of this discussion, you can give extra feedback or whatever you want to add to your students. More than that, please give. And if you want to give a feedback to me, I'm always ready, and I like that, because it is not the end. Tomorrow if I am giving to some more people or some more colleagues. I should improve my presentation skills and everything. So I'm open to you guys. I'm going for mute. Please unmute yourself and speak if you have any doubts. Professors and principals, whoever was there in the discussion, honorable lecturers, please, add your thought process also, so that it will help to me.
Dear students, if you have any doubts, please clarify with sir.
So nobody having any doubts? Okay. This is my last slide. As the corona situation is very severe, safety first. When you go to the industry or when you go to your college lab, always safety first. Whatever the experiment you do, whatever the chemicals which you handle, always be safe. Safety is the first thing. Industries also are like, how the pharmaceutical colleges are doing safety first. Industries also having first safety first principle. Corona, since the COVID-19 is pandemic situation, use sanitizer, wear face mask, maintain social distancing 6 feet or 2 meter, and wear mask all the time, and avoid multiple contacts, and go for the vaccination. And as a pharmaceutical students, it is our responsibility to explain the science involved in the vaccine. We know, right? We should not believe some stupid statements which there were some WhatsApp messages that after you take the vaccine you will die. That is not the case, right? Vaccine development is a pharmaceutical. There was so much of scenarios involved, and we know how much we will care of when we manufacture a vaccine. It's an entire science is involved, not some unnecessary thing. So you have to give awareness to your parents as well as your friends and non-pharmacy people, and your family members and friends and well-wishers, and encourage the vaccination. And thank you.
Thank you, sir. Thank you, sir.
Yeah, if you have any doubts, please clarify with sir, ma, or drop a mail in the chat box, ma. Please, anyone. So thank you so much, sir, for your kind information. And we would like to thank event directors, event management, and also a special thanks to Amarnath Reddy, sir, for giving such beautiful, wonderful information regarding the development of sterile isolation products. And once again, I'm thanking each and every participant for attending this wonderful webinar. Thank you so much.
Thank you. Thank you for your kind words, and looking forward to meet again. Thank you.
Thank you, sir.